Hello, my name is Afag Rzayeva. I am from Baku Engineering University. Today I am going to speak about the DNA extraction from banana. In this work, I will be working with my students, with Afag Rzayeva and Akshin Sarkiv. Why DNA molecules are so important and why we are going to learn this type of the molecules? Because all living organisms, human body, fruits, vegetables, animals, plants, they are made of a number of cells. And each cell carries information about whole organism. And DNA molecule have special component. We call these components as genes. DNA molecule carries information. How this organism must grow, how this organism must develop, and how this organism must have some features or with the lack of some features. These all features, characteristics, morphological, physiological characteristics are mainly determined by the presence of special genes, and these genes are located in DNA molecules. DNA molecules can be extracted from fruits, from vegetables, right? But today we are going to use banana. Why banana? Because banana is a soft fruit. And for this purpose, we can also use strawberry and other fruits. But in this experiment, we are going to use bananas. What we need for this experiment? Let's have a look to materials. First, we need peeled ripe bananas first. We need distilled water, 100 milliliter distilled water. We need 10 grams of sodium chloride, normal salt that we use in our daily life. We also use special protease drugs. Okay, and we also need some dishwashing liquid, about 10 milliliter. Approximately, we need 10 milliliter of dishwashing liquid. We also need alcohol for the extraction of DNA molecules. Firstly, let's have a look why we are going to use dishwashing water, why we are going to use these protease drugs, and what is the main purpose of the usage of the salt in this experiment. Firstly, dishwashing liquid helps to disturb the structure of special cell walls. And by this way, of course, DNA molecules can be extracted from the cellular environment. By this way, they can be extracted to the special liquid, to this dishwashing solvent. And of course, by this way, we can see DNA molecules. We need to see some DNA molecules with our naked eye, without any microscope, without any hand lens. And so this will be interesting. I hope it will be interesting and useful work for you, and you will study a lot about the DNA molecule today. Okay, let's begin to our work. Today we are going to take half of banana. Let's crush. Sarah, please crush this banana. Okay, we need to crush this fruit. Why we need? Because for the extraction, we have to destroy the structure of the cells and to destroy the structure of special tissues. You can add some water. Here we have got 100 milliliter distilled water. We can add some of them for a better crushing of these bananas. We must make something like a pudding-like structure, special smoothie solvent for a better extraction. If you need, you can add some much more water. Okay. crash this crash this solution, right? We have to make something like pudding, like smoothie. Okay, it's okay. Please pay attention. Here we have got bananas. Here we have got pudding, like the concentration of bananas with the water, right? We must pour this mix to the water. Here we have got 100 milliliter of water. Some of this water I add to this mixture. Let's pour this mix to the water. And with the help of the glass stick, we can mix this mixture. We must mix them for 5 or 10 minutes, depending on the concentration of the banana. Okay, thank you, Akshin. That's okay. Thank you. Here we have this mixture made of bananas and water. Now I'm going to add some dishwashing liquid. Here I have about 10 milliliter of this type of dishwashing liquid. Please, when you pour this dishwashing liquid, please be very, very careful here. That's okay. This will be enough. Okay, please mix them. Why we are using this dishwashing liquid? Because they contain special components, and these components can disrupt the cell wall. By this way, we can extract DNA from the intercellular environment to the extracellular environment. Okay, it's good. Let's add some protease. That's for a better extraction of DNA molecules. Here we have got some protease, and please take these drugs, 2 of them, very small drugs, and we must add these drugs to this mixture solution. And mix them. When you mix, please avoid the formation of foams. Okay, now let's add some salt, sodium chloride, to this mixture. That's okay. Mix them. Okay, thank you. Now let's filter this solution to this beaker. That's the special beaker, plastic beaker. And you must use here special filters here. If you haven't this type of the filter, you can just use normal coffee filters instead of these chemical filters. Please pour this solution. Okay, be very careful, and let's wait the filtration of this solution. This can take 10 minutes or 20 minutes. Let me check. Okay, it's okay. Let's wait. After filtration this solution, we must add some alcohol. In this stage, we must be very careful and we must use special frozen alcohol. And here, please mind here, you can also see DNA molecules, right? Yes, here. After 5 minutes, we can easily observe these molecules. With naked eyes, without any microscopes. The size of DNA molecules, the size is very small, but there are a lot of cells in our organism, as also in one fruit, also especially in banana, there are a number of hundreds, thousands of cells, and each cell have DNA part. And these all DNAs together, and the DNAs that we could extract from these cells, and the cells that we could disrupt by using special methods, by using proteins, we can observe this part of DNA molecules here. Here on the surface of this, please mind here. Yes, white part, they are DNA molecules. Yes, right, some special balance, right. Yes. By using special stick, we also can take this part of the DNA. Let's try to take this stick and try to touch some DNA molecules together. Okay, here you can see DNA molecules here, called frozen alcohol, could extract these DNA molecules. Sarah action, please, by using special anatomic needles, you can also take some DNA molecules. Like, interesting, right? That's the DNA. The part that contains a lot of information about the features, about the morphology, about the growth, about the development, and some information about the evolution of all living organisms on the earth. Okay, thank you for your attention.